Imagine going to the pharmacy right now. You pick up a prescription for a massive blockbuster medication. Maybe it's a weight loss drug, or maybe it's for your blood pressure.
Right.
You take it home, you follow the instructions perfectly, and, well, nothing happens. Or worse, you get terrible side effects.
Yeah, which happens way more often than people realize.
Exactly. And then you find out that just because your grandmother was born in Manila instead of London, that specific pill was essentially doomed to fail in your body from the very start. Like, have you ever considered that the medical treatment you're receiving might have been designed for someone with a completely different genetic background?
It is a really jarring thought. I mean, we are so conditioned to view medicine as universal. Like a broken bone is a broken bone. A bacterial infection is a bacterial infection.
Right, you expect it to be like engineering. You look at an x-ray, you see the jagged white line, and the doctor points to it and says, broken or not broken.
Yeah, but today we are going on a deep dive into a piece of research that proves the human body is absolutely not a standardized machine.
Not at all.
For decades, the math behind what we call precision medicine has been keeping this massive secret. It's been relying on data that really only represents a fraction of the global population.
So today we are looking at a groundbreaking 2026 article published in the journal Cell by Haas et al. It's titled Advancing Precision Health Discovery in a Genetically Diverse Health System.
And to really understand how this paper is just entirely rewriting the rules of medicine, we have to look at the engine driving these discoveries. And that's the UCLA ATLAS Biobank.
Right.
This isn't just another database. I mean, it is a fundamental shift in how we observe human biology.
So our mission today is to explore this massive project. The researchers managed to link the highly detailed genetic data of nearly 94,000 diverse individuals in Los Angeles directly to their electronic health records.
Yeah, and we're going to unpack how this incredibly rich data set is dragging precision medicine out of the muddy waters, really, and making it truly precise for you, regardless of where your ancestors came from.
Yeah, so let's start with the sheer scale of the blueprint here. Before we can get to the medical breakthroughs like the new disease risks and the drug interactions, we need to explain how they actually built this engine.
We are talking about 93,936 participants.
It's massive.
It is. And this data collection is incredibly rigorous. So they have 92,164 people mapped out with array genotyping, and then 61,797 with whole exome sequencing.
Okay, let's break those two terms down for the listener because they are crucial to understand. Array genotyping is essentially scanning the genome for specific known variants, right?
Exactly.
Like if your DNA is a giant encyclopedia, array genotyping is like using CO+F to search the book for a few thousand specific typos that scientists already know cause problems.
That's a really great way to visualize it. It's fast, but it only looks for what it already knows is there.
Right.
You can't find anything new.
Right.
Whole exome sequencing, on the other hand, is much deeper. So the exome makes up the protein coding regions of your genes.
Which are the parts of the genome that actually give the instructions to build your body, right?
Exactly. They are the instruction manuals. So sequencing the exome means they are reading those critical instruction manuals letter by letter.
So they aren't just searching for known typos. They're actually reading the text to see if there are entirely new typos we've never seen before.
Right.
But the genetic data alone is just half the equation here. To me, the real superpower of the ATLAS Biobank is the linkage to electronic health records, or EHRs.
Oh, absolutely.
These records go all the way back to 2013, encompassing over 71 million individual lab tests. Plus, they tracked 12,253 distinct fake codes.
Yeah, and a fake code is essentially a biological translator.
How so?
Well, think about how a hospital handles billing. They use ICD codes, which are, frankly, incredibly messy and redundant because they are designed for insurance payouts, not scientific research. So a doctor might use 5 different billing codes over the course of a year to describe a single patient's type 2 diabetes, just depending on the specific visit. A FILA code aggregates all those messy administrative codes into one clean, unified biological disease classification.
Wow, which totally changes the way you can track a patient's health.
Completely.
Because in the past, genetic studies usually relied on a really small, mostly uniform group of people, and researchers only checked their health at one specific moment in time. It was like looking at a blurry black and white snapshot. You might catch a shadow of a disease, but you miss all the context.
Yeah, but the ATLAS Biobank, by linking that deep DNA sequencing to those unified FELA codes and 71 million lab tests over more than a decade, it gives us something else entirely. It's a continuous longitudinal view.
It is like watching a 4K full-color documentary of a patient's entire life.
Right.
You see the exact moment the disease arrives, you see how the specific lab tests fluctuate week by week, and you watch how the medications actually perform over a decade.
Exactly.
But I want to push on something here. The researchers chose to do this entirely within a single health system, UCLA Health. To play devil's advocate, wouldn't pulling data from 50 different hospital systems across the country give you a larger, a better data set?
Well, you would get more volume, certainly, but you would introduce massive amounts of what researchers call confounding variables.
Okay, what does that mean in practice?
Imagine tracking blood pressure across 50 hospitals. Hospital A uses one brand of automated cuff. Hospital B does it manually with a stethoscope. Hospital C has different criteria for even admitting a patient. And Hospital D uses totally different software to record the numbers.
Oh, I see. So if you compare those patients, the differences you see in their health might just be differences in administrative protocol.
Exactly. The data gets incredibly noisy.
Right.
By keeping this entirely within the UCLA health system, they stripped away all that noise. Every single participant had their blood drawn in the same system, their labs were run through the same standardized machines, and their diagnoses were entered into the same software.
So it creates a pristine apples-to-apples comparison.
Yes. We no longer have to wonder if a data anomaly is just a typo from a billing clerk. We know it's a biological reality.
Okay, so let's pause and lock this in for you listening. Time for a quick recap. Our first major takeaway. The ATLA's biobank works because it marries deep genetic sequencing with over a decade of continuous medical records, all purified within a single hospital system to eliminate noisy data. It is the ultimate pristine tracking system.
And with that pristine tracking system in place, the researchers had to figure out how to categorize these 94,000 people to actually find genetic patterns.
Right, which brings us to the diversity of Los Angeles.
Yeah, the study initially breaks the populations down into 5 broad-scale continental ancestries. So you have European, African, East Asian, South Asian, and admixed American.
And just to clarify that last term, because we hear it a lot in genomic studies when the paper says admixed American, they are generally referring to populations with a complex mixture of indigenous American, European, and African ancestry, which is highly typical of Latin American populations.
Yes, exactly. But the researchers didn't stop at those 5 broad categories. They zoomed in significantly further, analyzing the DNA to identify 36 highly specific, fine-scale ancestry clusters.
And to do that, they use a mechanism called identity by descent, or IBD. And I really want to make sure we understand the mechanism here, because this is how we find hidden genetic relatives. How does IBD actually work on a molecular level?
So identity by descent looks for specific, long segments of DNA that are identical between 2 individuals. It's less like putting together a puzzle and more like finding a highly specific typo in 2 different copies of a book.
Okay, I like that.
If you and a total stranger both have the exact same incredibly unique typo on page 42 of a massive novel, it is highly unlikely you both made that typo independently.
Right.
It means you both bought your book from the exact same printing press.
You share a common source.
Exactly. When researchers find these long, unbroken blocks of matching DNA in 2 people, it proves they inherited that exact chunk of DNA from the same recent ancestor like a great-great-grandparent.
Wow.
By mapping these shared IBD segments across all 94,000 people, the algorithms mathematically grouped people into those 36 fine-scale clusters based on very recent demographic history.
Okay, let's unpack this. Are we effectively redefining what race means in a doctor's office? Because traditionally, you walk in, you get a clipboard, and you check a blunt box on an intake form. White, black, Asian, Hispanic.
We are absolutely moving away from social constructs and toward biological realities. Those broad census categories on the intake form are socially important, sure, but biologically they swallow up incredibly distinct groups.
Right.
A category like Asian encompasses billions of people with vastly different evolutionary and genetic histories.
And the findings that drop out of these fine-scale clusters absolutely prove your point. For instance, because they use this IBD mapping, the ATLA's biobank now contains the largest genetic data set of Filipino individuals ever compiled for research, 1,438 people.
Yeah, it's unprecedented.
And they found disease risks we never knew about. Filipino participants had the lowest risk for vitamin B complex deficiency compared to everyone else. But they had a massive 4.3 times higher risk for cholesterolosis of the gallbladder.
Yeah, and to explain the mechanism there, cholesterolosis is a condition where Macrophages, these immune cells that basically act like tiny garbage trucks in your body, they start engulfing excess cholesterol and they get stuck on the wall of the gallbladder.
Yikes.
Yeah, it causes a lot of inflammation and pain.
So a Filipino patient and an East Asian patient might check the exact same box on an intake form, but their biological risk for their gallbladder filling up with cholesterol is totally different.
That's the danger of the blunt checkbox. And the researchers found another cluster, Iranian Jewish participants, who showed a significantly elevated risk for glaucoma.
Which is an eye issue, right?
Right. It's a condition where pressure builds up in the eye and damages the optic nerve. That specific genetic risk was completely unreported before this study, simply because Iranian Jewish individuals are usually lumped into a broad white or Middle Eastern category in medical data.
So they just get lost in the noise.
Exactly.
Okay, time for another quick recap for you listening. Takeaway 2. Broad racial categories hide specific biological dangers. By looking at identical blocks of shared DNA, that identity by descent mechanism researchers can group us into fine-scale ancestries, revealing severe localized disease risks like gallbladder disease or glaucoma that standard medical forms completely miss.
Which naturally forces us to look at a massive mathematical problem in modern medicine.
Oh boy.
If different ancestries have these highly specific risks, what happens when doctors try to use traditional genetic tools to predict who is going to get sick?
It usually ends badly. We need to talk about polygenic scores, or PGS. If you are listening to this right now and your heritage isn't strictly Northern European, your doctor might be looking at your genetic risk factors through a heavily distorted lens.
Yeah, polygenic score is a predictive tool. Most common diseases like heart disease or diabetes aren't caused by one single broken gene. They're caused by tiny compounding variations across thousands of different genes. So a polygenic score adds up all those tiny variations to give you a single number that predicts your overall risk for developing a specific disease.
It's essentially a credit score for your health. If your score is high, your doctor knows to watch your heart very closely. In theory, it's revolutionary.
In theory, yes.
But the ATLAS data exposes a severe mathematical flaw. The researchers proved that these polygenic scores are incredibly robust and accurate if you are of European descent. But the moment you apply those same scores to non-European ancestries, their effectiveness is, quote, markedly diminished.
The math literally does not work.
Yeah.
And the reason why is deeply rooted in how the databases were actually built. For the last 20 years, the massive genetic databases used to calculate those polygenic scores were built almost entirely using the DNA of people with European ancestry.
It's exactly like trying to use a street map of London to navigate Tokyo. The map itself isn't broken. It's a perfectly good, highly detailed map of London. But if you try to use it in Tokyo, you are going to crash into a wall.
That is a perfect analogy. And in a clinical setting, crashing into a wall means patient harm. As polygenic scores are pushed into real-world clinics, relying on these Eurocentric databases is actually dangerous.
Because it gives false confidence.
Exactly. You might calculate a score for an African-American patient and tell them they are at low risk for peripheral vascular disease, entirely missing the fact that the algorithm simply doesn't know how to read their specific genetic map.
So how do you actually fix that? If the map is biased, how did the UCLA team solve it?
Well, they used advanced computational predictors. They essentially fed their diverse ATLAS data into the algorithms and retrained them to look for disease gene links within the diverse populations themselves. They forced the math to account for the different genetic backgrounds.
And the moment they did that, the blinders came off. They uncovered entirely new, unreported links between specific genes and diseases.
They're really good.
For example, they found a strong association between a gene called ANKZF1 and peripheral vascular disease, specifically in African Americans. Yep. Now, peripheral vascular disease is a condition where narrowed blood vessels reduce blood flow to the limbs, causing severe pain. Previously, that specific genetic warning sign was totally invisible.
They also linked a gene called FN3K to intestinal desaccharидase deficiency in Europeans and admixed Americans.
That's quite a mouthful. What does that mean for the patient?
It's a condition where your gut lacks the specific enzymes needed to break down complex sugars, leading to severe digestive issues. So suddenly, with the retrained math, we can see these risks.
Wow. So quick recap number 3. Polygenic scores, the credit scores for your health, have historically failed anyone who isn't of European descent because the baseline data was just too narrow. By retraining the algorithms on the diverse ATLAS Biobank, researchers are finally finding the missing genetic warning signs for vascular and digestive diseases across all populations.
And it proves that greater ancestral diversity in clinical research isn't just a moral imperative, it is a strict, undeniable mathematical requirement for safe medicine. You simply cannot practice precision medicine if your tools are imprecise for the vast majority of the global population.
That brings us to the final and perhaps most mind-blowing part of this deep dive. Predicting disease is one thing. Saying you are at high risk for vascular disease is theoretical.
Right.
But the ATLAS study didn't stop at prediction. They took this diverse genetic data and applied it to a patient taking an actual physical drug today. They moved from predicting disease to actively treating it.
They chose one of the most famous, widely discussed medications in the world as their test case, semaglutide.
Semaglutide is the active ingredient in blockbuster drugs like Ozempic and Wegovy. It is an absolute juggernaut right now for weight loss and type 2 diabetes management. Literally everyone knows someone taking it.
It is a generational drug. But crucially, it doesn't work perfectly for everyone. Some people lose 20% of their body weight, while others barely lose anything at all.
Here is where that 4K video documentary of the patients' lives really flexes its muscles. The researchers could track patients who were prescribed semaglutide, watch their exact weight loss progress month by month in the medical records, and then cross-reference that physical progress with their specific genetic ancestry.
And what they found changes how we think about pharmacology. They discovered that the amount of weight a patient loses on semaglutide varies significantly across different genetic ancestries.
Okay, hold on. I want to push back on this a bit. Semaglutide was engineered in a lab. It's a synthetic chemical. Why on earth would a lab-created chemical care about where my great-grandparents lived? How does ancestry actually change how a pill works?
Because the chemical doesn't operate in a vacuum. It has to bind to biological receptors in your body to work. And your genes act as the blueprint that builds those receptors.
Oh, I see.
The study found that the efficacy of semaglutide is heavily modulated by genetic variations in a specific gene called PTPRU.
Let's explain PTPRU. What does it actually do?
Well, PTPRU is a gene involved in cellular signaling. It helps dictate how cells communicate with each other and how they receive outside signals.
Like docking stations.
Exactly. If your specific ancestry gave you a variation of the PTPRU gene that slightly alters those cellular docking stations, semaglutide might literally have trouble binding to your cells the way it was engineered to.
It is wild that a synthetic drug doesn't behave the same way in two human bodies purely because of the microscopic code written in their DNA.
This is the ultimate promise of pharmacogenomics. Pharma, meaning drugs, and genomics, meaning your DNA. It's the study of how your genetic map affects your response to medications.
Right.
Right now, a lot of medicine is still trial and error.
Yeah.
You try an antidepressant, you wait 6 weeks, you see if it works. If it doesn't, you start over. You try a blood pressure med, and if it makes you dizzy, the doctor tries another one.
It's exhausting, it's expensive, and for the patient, it's incredibly frustrating.
But the ATLAS Biobank points to a near future where a doctor can look at your fine-scale genetic map, check your PTPRU variation, and say, we aren't going to guess. Semaglutide won't be effective for your specific cellular receptors, but based on your DNA, this other medication will work perfectly.
Knowing exactly which drug will work for you before you even swallow a pill. Okay, so let's distill this entire journey. What does this all mean for you listening? We started by shattering the illusion of precision medicine. We thought we had a perfect map of human health when really we only had a map of a very specific, mostly European demographic.
Yeah, the UCLA ATLAS Biobank proves that precision medicine without diversity is just an illusion.
By building a biobank of nearly 94,000 diverse individuals within a single health system and combining their DNA with massive longitudinal health records, researchers have created that 4K documentary of human health. They showed us that by breaking populations down into 36 fine-scale genetic ancestries, we can find hidden disease risks, like gallbladder risks in Filipinos or glaucoma in Iranian Jewish populations.
And we learned that relying on old polygenic scores is dangerous, and that by retraining the math, we uncover entirely new gene disease links, like ANKZF1 and vascular disease.
And finally, we saw genetics in motion. We learned that everything from your baseline disease risk to exactly how much weight you'll lose on a blockbuster drug like semaglutide depends entirely on your unique genetic heritage, right down to the PTPRU gene. It fundamentally changes how we view treatment.
It really does. But it also leaves us with a pretty provocative question.
What's that?
If our genes dictate exactly how well blockbuster drugs work, If your DNA is the literal key to unlocking the right medication, are we approaching a future where getting a prescription requires a mandatory DNA swap?
Wow.
And if we do reach that point, what happens to the populations around the world who are still left out of these global biobanks?
A heavy but absolutely necessary thought to leave on. We want to thank you for joining us on this deep dive. Keep questioning the world around you. And remember, the next time someone tries to hand you a medical x-ray or a genetic test that looks perfectly clean and simple, make sure you ask who the machine was calibrated for.